Hello, this is Danu, and today we are going to discuss about brain developments from the baby state. Let's get started. So, when we are babies inside our mom, the first step in our baby brain development is cell division. Another key step is cell differentiation, in which individual cells stop dividing and take on specific characteristics, such as neurons or glial cells, and glial cells are just another neural cells that assist the neurons. Differentiation orders things spatially. Different kinds of neurons migrate to various locations in a process, and it is called pattern formation. The first major event of pattern formation takes place in the third week of human gestation, when the embryo is just two connected sheets of dividing cells. A small patch of cells on the upper surface of bilayer is instructed to make the entire brain and spinal cord. These cells form a tennis racket-shaped structure called the neural plate, the front of which is destined to form the brain, the rear to be the spinal cord. Signals directing the destiny of these cells come from the layer beneath that goes on to form the midline skeleton and muscles of the embryo, which is called mesoderm. Various regions of the early nervous system express different subsets of genes, presaging the emergence of brain areas: forebrain, midbrain, and hindbrain. So in this illustration, A represents the human embryo at 3 weeks after conception, which we just talked about. B talks about the neural plate forming the top surface of the embryo. C talks about a few days later, when the embryo develops enlarged head folds at the front end. And DEF illustrates the different levels of the axis from head to tail, showing various stages in neural tube closure. So a week later, the neural plate rolls up, closes into a tube, and sinks into the embryo, like the middle illustration shows. In other words, initially, the neural tube develops with the genes of our skin cells, and eventually the neural tube, or the future spinal cord and brain part, sinks into our skin. So it becomes enveloped by the future epidermis, which is another word for our skin. Further changes happen in the next few weeks, such as changes in cell shape, division and migration, and cell-cell adhesion. For example, the neural tube flexes such that the head region is bent at right angles to the trunk region. This patterning progress leads to finer levels of resolution, ultimately conferring individual identity to the young neurons. This is just another model of the above models, showing the different parts of the baby brain and neural nervous system. There is forebrain, midbrain, hindbrain, and as the time goes on to 3 weeks, 4 weeks, 3 months, and 6 months, the brain begins to form and begins to look like the thing that we know now. Now let's talk about the different names of brain parts from its initial development stage to the growing stage. So initially, in the 3 to 4 week embryo that we just talked about in the previous section, the brain part looking somewhat like A here, there are 3 primary brain parts. Here, cephalon means, cephalus is a Greek word for head, and it is used to identify the different brain parts. There are 3 primary brain parts in the initial stage from 3 weeks to 4 week embryo. There are prosencephalon, mesencephalon, and rhombencephalon. Prosencephalon got its name from the fact that it is in the very beginning, in the very front of the brain part. As you can see in the lateral view here, it is in the very front. Because pro means in the very front, it is named that way. Mesencephalon got its name from the fact that meso means mid, and the word that attaches meso and cephalon looks like mesencephalon, so it got its name from there. Rhombencephalon got its name from the fact that it not now, but it later would look like a rhombus-like structure when it develops later. Now, prosencephalon divides into two sections, telecephalon and diencephalon. Tele means far away, so the fact that this part of the brain is far away from the rest of them named this part telecephalon. And diencephalon, di means two or second, so this is the second part of the brain, so it got its name diencephalon. Mesencephalon is the same as before. Rhombencephalon divides into metencephalon, myelencephalon. Metencephalon got its name from the root word meta, which means change. Meta plus cephalon became metencephalon. And myelencephalon got its name from the fact that it later develops to protect the spinal cord, and myelin is a word that represents protection. These different initial brain parts develop into the actual brain parts, like cerebrum, eyes, thalamus, hypothalamus, epithalamus, midbrain, pons, cerebellum, and medulla oblongata. And they later develop into a complex nervous system with neurons that communicate with each other to survive and live well. This is the end of this video, and thank you for watching. Bye!